What we're going to look at next is the liver, and then we'll take a peek at the gallbladder. We actually don't have good slides of the gallbladder on the digital slide, so I'm probably not going to hold you to that one, but I'll at least show you what it looks like. So the liver, we're going to see, is one of the largest internal organs of the digestive tract, usually upper right quadrant area. Again, going across the center line a little bit. Involved highly in a lot of metabolism of a number of different things. So involved in carbohydrate, lipid, and protein metabolism, stores glycogen, produces a lot of different blood proteins, so our clotting proteins, our blood albumins. In terms of digestion, it makes what is called bile, which is actually part of a byproduct of the breakdown of hemoglobin from blood cells that have got older. Some of the byproducts in that are in that bile, but what bile is doing is it emulsifies fats, and what that means is it reduces the surface tension of fats for one another. Fat and water do not really care for each other. Lipids and water, they tend to separate. By doing this, it takes what would become one big fat glob and turns into a bunch of little ones that lets the lipases and some of those other enzymes that I mentioned get in there and actually go after them. The other thing the liver does a lot of detoxification. It has 4 main lobes to it. Overall 2 major lobes, and then there's a couple of smaller lobes underneath. And again, all the blood from this is coming from the digestive tract up through what's called the hepatic portal system. And we're going to see that there's branches of that are going to feed these main, the main liver structures that we have, these little lobules. So you're going to have these hepatic portal vein, piece of that, hepatic artery, and then like some of these other ducts all entering into these main areas. So you can see you have hepatic artery bringing oxygenated blood in, portal veins bringing deoxygenated, nutrient-rich blood from the intestines. There's going to be one main hepatic vein leaving, and then the bile ducts that are coming out of the bottom that go to the rest of the digestive tract. And you can see some of this blood flow here, the main hepatic artery. You can see the portal vein is actually much larger, so it's all your blood flow from almost your entire digestive tract, with the exception of a little bit of the rectum, are going to be taking blood back to the liver. The cells of the liver are called hepatocytes, and like I said, they're arranged along these blood vessels like little bricks. We're going to see a lot of connective tissue in here holding this together, with a lot of reticular fibers holding these in place. And we're going to see that these capillaries that are going through the liver are actually those sinusoidal type, so very open capillaries that let us do a lot of stuff with this blood. Hepatocytes, like I said, polyhedral cells, mainly cuboidal though. I mean, when you really look at them, lots of endoplasmic reticulum, both smooth and rough, and sometimes we'll see glycogen granules if we really looked for it in there. Again, what these are doing, making a lot of the blood proteins that we're going to need. They secrete some of the acids that are part of that bile, actually also helping recover quite a bit of that one. They're taking the bilirubin, which is part of the breakdown of hemoglobin, and breaking that down, as well as detoxifying drugs, storing glycogen, storing fat, and being involved in the process of making glucose from other substances called gluconeogenesis. You can kind of see an example of a liver cell right here, a hepatocyte. You can see large amounts of rough ER and smooth ER in this image right here. Again, what is it doing? A lot of synthesis, bile either recirculating or generating these stuff from other byproducts. When we really start looking at the histology liver, we're going to see it's into these kind of hexagonal shaped lobules that are going to be filtering the blood that is moving to a central vein in the middle of these ones. And we have what are called these portal triads on the outside, which is a branch of the hepatic portal vein, a branch of the hepatic artery, and a bile duct or the periphery. So you can see that portal triad right here. And again, kind of showing you that this is a lot of branching. And if you really look at it, it does start looking like a filtering type material when you see an image like this one. And that's really one of the things the liver is doing, is acting as another one of our filters that is really monitoring what's in the blood and being able to do a lot of stuff with the blood components. So we can see a zoomed out low power one here. You can kind of see it's pointing at the center with this arrow right here, pointing at the edge right here. If we zoom in a little bit more, you can kind of see that basic shape right here, that hexagon shape, central vein. You can see a portal triad right over here. So this shows you much more so that central vein. So all this blood has to work from the outside of that hexagon into the central vein. Along the way, we have all these little sinusoidal capillaries that are allowing the liver to modify what is in that blood. You can see a picture of the portal triad here, the hepatic artery, branch of the hepatic portal vein. Again, you can see the difference in the thickness of the walls as well as the bile duct there. And what we have going on here is we have blood flow working from the outside in on these ones. So you have the portal vein and the hepatic artery that are both going to be leading blood flow from the outside of these sinusoids to the center, going to that central vein, which is then those parts of that vein are going to go from these different lobes and form together to give you the hepatic the hepatic vein, which is then going to merge into the inferior vena cava just below the heart. So this is kind of showing you that right here. You can see a branch of the hepatic portal vein, blood would be going this direction. Branch of the hepatic artery, blood is going this direction. We have a bile duct or canal going this, the flow on that one would be actually going the opposite direction towards the outside, and you can see the bile duct right here. They don't really draw it on this one, but the central vein would be in the center there, and like I said, all that flow is moving along these sinusoidal capillaries, letting the liver modify what is in the blood as it works its way to the central vein. You can see a little bit better on this one. So you can see how it's just a bunch of sinusoids, again, everything working to the center, allowing the liver to modify what is in this blood. The flow is going that way for the blood. You can see another image of it here with these sinusoids, kind of showing you more of a three-dimensional, how those sinusoids look. Like I said, bile, which is the main component of digestive secretion of the liver, these are actually going to be moving the opposite direction. So we have these bile canaliculi that are surrounded by pretty much two cuboidal cells on cuboidal cell on either side, these hepatocytes. Bile is going to flow from the center of these sinusoids out to the edge to the bile duct, and then those bile ducts are going to, we'll see sooner or later, merge together, and you're going to get two main left and right hepatic duct of bile that is then going to start going to what we refer to as the biliary apparatus, which is what's moving all that bile around and down to the duodenum. So like I said, these sinusoidal capillaries, incredibly leaky, discontinuous basement membrane. We have what are called these resident macrophages called Kupffer cells present in there. And again, lots of arterial and venous blood mixing together in here, allowing the liver to really secrete a lot of things into this blood as well as take a lot of things out of this, and the liver's getting first pass. Again, that everything that's cut, with the exception of some of the dietary fats which went to those lacteals, is getting first pass at all that blood that's coming back from the digestive tract. Underneath those sinusoidal capillaries is we have what is called the perisinusoidal space, or the space of Disse. Again, another one of those funky names that we get in histology, the space of Disse. It's that space between the sinusoidal capillaries and the hepatocytes. Decent amount of microvilli in there, allowing on these cells that are lining that really allow us to absorb and modify a lot of the stuff that's in there. Lots of exchange of nutrients and other materials are taking place as we're taking stuff out of this space of Disse or secreting things into it. And this is showing you the Kupffer cells, the mid-liver macrophages being stained dark. So these are resident macrophages of the liver that are going to be, again, anything that might have got back from the digestive tract, getting in there and being able to trigger immune responses on that stuff. So the last thing I want to kind of talk about here is the gallbladder. So all the bile that is made by the liver is going to be passed down through a couple of main ducts, and we're going to see it's actually stored and concentrated in the gallbladder. So you can see, this is what I was saying before, the left and right hepatic ducts. This becomes the common hepatic duct, which is taking the bile down to the gallbladder. You can see it can go up this cystic duct right here, and then we can store that bile in the gallbladder. If the CCK is released, that gallbladder can contract, and it will actually pump all this down what is called the common bile duct into the duodenum, and that again is going to help us emulsify those digestive fats, make them into smaller droplets so those lipases can get after them. So again, the gallbladder stores and concentrates the bile, And then under signal, it will actually contract and release that bile out. If gallbladder is removed, the liver can actually just release the bile right down to the duodenum. Most of the time that's not a problem, but again, they usually say, especially right after you've had surgery for that, a lot of times they are really going to try to say limit your dietary intake of large amounts of fat, so don't eat a super like cheeseburger, fries, and a chocolate shake type of thing right after you get your gallbladder out. It would not be pretty. CCK, like I said, is what is actually going to trigger the contraction of that, and we know it also works on the pancreas to release some of its secretions. So you can kind of see how that's a signal to get all the ingredients into that mixing bowl, which is the duodenum, so we can start doing the rest of the digestive functions. And you can see these bile canaliculi and the bile duct here within the liver. Again, and this is the basic bile flow out of the liver over to the cystic duct and into the gallbladder, and then you can see common bile duct down to the duodenum. So really, to me, on this stuff, we're going to take a real quick look at the liver when we're all done with this. And then, like I said, I don't think there's actually a gallbladder side. I'm not planning on having you do the gallbladder, but understand bile flow and what the gallbladder does. You can see a picture of it right here, zoomed out, a little bit more zoomed in. One of the things you can see on the gallbladder is that it has quite a thick muscularis. It is a muscular organ. A lot of times if you get gallstones, that's what it's squeezing on and causes the pain. But you can see quite a big muscularis layer on these, and pretty much a simple columnar epithelium on the interior. So again, like I said, we'll look at the sides of the liver, and then that will be it for this chapter. You can see a little bit more of the muscularis, the serosa. The side we're looking at here is a liver gallbladder side. If you look in the upper corner, you can see the gallbladders in the center of it with a big open space, and then you can see the liver around it. We are in the upper region up here. The main thing I want to point out is just kind of some of the parts of one of these sinusoids here. So you can see where I have the cursor right here is where that central canal would be, and like I said, they're mainly kind of a hexagonal shape that goes around that. Again, flow is working from, this would be a portal triad right there, which we can zoom in on a little bit more. But you can see there, those three structures. That would be the bile duct, there's the artery, there's the vein, the different parts of that right there. The flow is working from these towards that central canal through these hepatic sinusoids. And again, nothing in here, this is just an H&E stain, nothing stained for anything in particular on these ones, but again, knowing that this is liver, knowing the central vein, the different portal triads and what the part of that are, it was the stuff that I would say would be fair game. If we actually now move and take a look at the wall of the gallbladder, you can see that simple columnar epithelium, the underlying submucosa, and then that really thick layer of muscularis right there.